so I didn't stuff up over keys again, and to describe ones new to science, and eventually to produce a key. But to do this, I needed some tools. The first one was to have some collections, and so I've been making collections over the past 30 or so years. And being associated with the Western Australian Herbarium means that I have access to herbarium collections from, well, not just Western Australia, but in Australia and around the world. Secondly, I needed to have a system for making consistent records. And I've used the methodology developed by Rod Tullis in New Jersey, which is very detailed, but it means that you don't forget anything. And I've used this for both macroscopic and microscopic characters. I realized, of course, that with any new species descriptions, you needed to have sequences. And I realized also that this was quite beyond my capability. And I've been very grateful to Lawton McGirt for spending a lot of time finding out how to extract DNA reliably from local Ammoniters, and also showing that it was necessary to clone to get clean ITS signatures. And more recently, Danielle Gostiano has taken over from Lawton, and she does all the sequencing. Danielle is a sessional academic at Curtin, so it has to fit around her teaching activities. And lastly, I found I needed to have time to do this, so I waited until retirement. Now, Ammoniters are easy to recognize in the field as a group, and this is because they have a universal veil. We have on the left Ammonita muscaria, which is a type of the genus. And you can see the universal veil because it's very clear. It's a different color from the color of the cap. The way the universal veil breaks up as the cap expands and the stipe elongates gives some indication as to the orientation of the hyphae and inflated cells in the universal veil. And in Ammonita muscaria, they're a bit like a crew cut over the top of the cap. And so as the cap expands, the universal veil breaks up into straight-sided warts over the surface. In Ammonita wood the universal veil is a much more felted layer, which expands to some extent as the cap expands. And so it doesn't break up into the very straight-sided warts that you see in Ammonita muscaria. Now, the universal veil originally is attached at the top of the bulb, which is at the base of the stipe. And this sometimes leaves ridges and warts, and sometimes a membrane at the base of the stipe. The universal veil used to be called the vulva, but the vulva is a term that's best restricted to a certain type of universal veil, which I'll show you later. So in addition to a universal veil, there is usually a basal bulb in Ammonites, but not in all species. There is usually a ring in Ammonita, but not in all species. And the majority of species have white or pale gills. But as you've seen already, one of the species has brown gills. So a bit about the genus. It's cosmopolitan. There are more than 1,000 species, and genetics has shown that it's monophyletic. Although the universal veil is a very useful field character, it's actually defined by the anatomy of the gills, by the anatomy of the stipe, and the schizoharmonial development of the gills. The genus is ecologically important because the majority of species are mycorrhizal with woody plants, although there are some saprophytes. And it's economically important because some species are edible and some are poisonous. And most mushroom poisonings are the result of Ammoniters. And in Australia, it's widespread and conspicuous. There are more than 100 names now, and there are different forms. The majority are agaricoid, so like mushrooms, but there are also sequestrate ones, such as the secotioid one in the center here, where you have a stipe and a cap, but the cap never expands properly, and so the spores are not dispersed in the way that they are from the mushroom. And there are some hypergea species too. Now, in a genus which is so large, it's natural to try and split it up in some way. And initially, this was done on macroscopic characters, but it seemed that they weren't really very consistent and it didn't really work. But about 100 years ago, it was realized that if you put spores into iodine, In some cases, they changed colour, they went blue, and in some cases they didn't. And I can assure you that in that drop of iodine on the left of your screen, there are spores there, but they just haven't changed colour. If you look under the microscope, you can see that the blue coloration is in the walls. And this amyloidy is considered to be a basal state because it's easier to lose this characteristic than to gain it. And this amyloid reaction was found to be useful also in predicting such features as differences in the margin of the cap. And it's been used to split Ammonita into subgenera, initially into 2, but now into 3. So here's an example which is perhaps relevant. We have 2 mushrooms here. The one on the left is Ammonita caesarea, which is an edible species that occurs in Europe. And on the right, we have Ammonita phalloides, a poisonous species that occurs in Europe. And when you look at them, they are actually fairly similar. Both of them have a membranous universal veil, which is a form that's called a vulva. And this remains at the base of the stipe. Sometimes you have a patch of membranous tissue on the cap as well. Both of them have yellowish coloured caps, but it's a little bit more orange in Ammonita caesarea. Both of them have a ring. Both of them have pale coloured gills. But if you mount spores of Ammonita caesarea in iodine, you find that they are inamyloid. If you mount spores of Ammonita phalloides in iodine, they are amyloid. So these are similar in appearance, but they're from different sections of the different subgenera in the genus. The most recent phylogeny of Ammonita is a molecular phylogeny which was published in 2018. It covers a very large range of species, and it's based on concatenated LSU, TEF1, RPB2, and beta-tubulin genes. And it divides the genus Ammonita into 3 subgenera. There's subgenus Lepidella, which has amyloid spores and is saprophytic. So it includes the saprophytic species. We have subgenus Ammonitina, which has amyloid spores and is mycorrhizal, and subgenus Ammonita with inamyloid spores and mycorrhizal. And the saprophytic habit, together with the amyloid spores, suggests that subgenus Lepidella is basal to the group. Now, the subgenus Ammonitina is the one that's most common in Australia. And this most recent phylogeny subdivided the subgenus into 6 sections. And you've seen some of these species already. We have the brown-gilled Ammonita ocroteria in section Rhinocensis. We have Ammonita phalloides, the deadly poisonous one, in section Phalloideae, which is the death cap mushroom group. And then we have section Arenariae, which includes Ammonita wadalawitu, which I found at Murdoch University many years ago, and section the species Ammonita arenaria, which is secotioid. And these are only known from southern Australia. So it's a section which is Gondwanan. In subgenus Ammonita, we have Ammonita muscaria in section Ammonita. We have section Caesarea, which includes Ammonita caesarea. And we also have section Amarendii, which includes the species Ammonita umbranella. And this section is only known from Australia and South America. So it's another Gondwanan section. And this, I think, is really interesting because it suggests that the genus has diversified once Gondwana had broken up. And there may be other Gondwanan sections which are yet to be discovered. Now, the money from AMS is being used to look at Ammonita umbranella. And I think it's interesting because it's this Gondwanan from this Gondwanan section. It's a species which was described originally from New South Wales. but it's been a bit of a catch-all name for several species which have a brown cap and have inamyloid spores. In Western Australia, we have, well, what's called Ammoniter umbranella, but it's not one species. There's at least 2 species, and I'm showing these 2 here. We have, both of them have brown caps. Both of them look fairly similar. They're large, they're quite common. But if you look at the ring, the one at the top, the ring sticks out more or less at right angles to the stipe. The one at the bottom is much more pendulous. If you look at the spores, they are slightly different shape. And if you look at the sequences, they are different. And this is probably true for other species named as Ammoniter umbranella around Australia. So what I've done is try to source collections of Ammoniter umbranella, or something which is called umbranella, from collections from the state herbaria around Australia. I haven't asked them all. I've asked for ones which are perhaps less than 40 years old. And using the money from the AMS project, Danielle has tried to sequence all of these collections. So far, not with great success, but we've certainly got some sequences out of them. I've been plodding along with the micromorphology to try and see if they fall into groups. And the first thing I would do is to look to see whether they have amyloid or inamyloid spores, and not all of them have inamyloid spores, which I would expect from Ammoniter umbranella. So there's obviously a lot of mixed collections in there. So we were rather held up with this because last year the collections, the herbaria weren't material I've asked for. The last one arrived in about June. And we are nearly finished with the sequencing. I'm still working on the micromorphology. So to summarize this, Ammoniter is a cosmopolitan genus. It's very large and it's very conspicuous. The genus is easily recognized because of the presence of the universal grail, but the species are very difficult to separate without a combination of macroscopic, microscopic, and molecular characters. It's ecologically important because the majority of species are mycorrhizal. It's economically important because some are edible and some are toxic, and they are extremely common in the Australian bush. There are 2 sections in Ammoniter which have are of Gondwanan origin, and Ammoniter umbranella is really a catch-all name, which includes several species. And I guess we're just trying to sort them out using sequences from the AMS money and the morphology. So finally, some acknowledgements. Thank you to Danielle for doing the sequencing, to my husband Peter, who can spot Ammoniters at about 50 paces, to Lawton McGurk, who did the original work on extracting DNA and working on how to get reliable ITS sequences, the West Australian Herbarium for sourcing material from around Australia, and funding over the years from ABRS, from their non-salaried researcher fund, from the Western Australian Naturalist Club, from Lottery West, and most recently from AMS. Thank you very much.
Thank you so much, Elaine. That was really interesting. It's fantastic to see the research grant being put to such good use. You can stop sharing your screen if you just go to the top. I have a question to kick off with. First of all, your talk really highlighted to me the importance of maintaining expertise in morphology when it comes to speciating and identifying fungi. So I'm really excited to hear that, and in particular the use of amyloidy. I mean, in my limited experience, it's not something I'd come across it before. Is it something that is used to characterize different genera outside of Ammoniter?
Well, amyloid reaction is quite important in the spores of some species, such as Russulas and Bacterius, where the ornamentation is amyloid. And also there are some genera where you have a dextrinoid response in the spore. So it's a very useful character to use, if only to say there's no response.
Yes.
That might well eliminate some species.
Yeah, and I think it's probably also a lot quicker than doing sequencing in that sense. If you know whether or not something's going to have that reaction or not, it's probably one good way of confirming.
I think there are a lot of things where you can just look down a microscope and immediately eliminate some possibilities.
Yeah.
It's a skill that has tended to be overshadowed by more recent techniques.
I agree completely. David Holden works here with me, and his knowledge under with the microscope definitely speeds up the process a lot of the time. So it's great to have, those much faster techniques. I mean, there's all the talk about DNA and sequencing, but you can't do without the morphology, that's for sure. Where are you going from here then? So now that you've got all this information, what are you going to be doing next?
Well, we'll be putting it all together and seeing whether we can come up with a number of species in Ammonita umbellata, and it will just become part of the Ammonita work that I've been doing over such a long time. But I would really like to put together an Ammonita key based on sound morphological descriptions supported by DNA sequencing. And it would be really nice to run an Ammonita workshop. I'm not sure I'm up to running one in New Zealand, but I'd give it a go.
Well, if we have our conference next year in New Zealand as planned, that would be probably good timing. I'll give you a heads up. Tom May would like to ask a question. Go ahead, Tom.
Thanks. Hello, Elaine. Love your work with Ammonita. Some fabulous work to sort out some really difficult complexes. With this group, do you expect that you'll be able to recover morphological characters from the type specimen that will allow you to pin it to one of the species that you've identified? And would it be useful to be sequencing the types? And is that something that you might need to be, wouldn't it be great to have sequences from all the different types that are out there, but then that gets difficult because it's hard to get DNA out of it. So I guess the question is essentially about types and what to do with.
Well, in all of the material that I've borrowed, where there are type specimens, I'm not allowed to sequence types. That's, I think, a prohibition by many herbaria. And I don't think I want to, because the techniques we have are really not adequate for working with really old material. So I think that's something which would be better off done by others who have access to different sequencing techniques. So I've just been looking at the morphology of types, and sometimes it's quite useful because you can eliminate, you can use spores or presence of camp connections or orientation of hyphae and so on and so forth, which might indicate whether or not you have, you can see similarity collect to have sequences from all the old types. But I don't think that's something I would be prepared to do. I think it needs to be done by herbaria, and it needs to be done using different techniques, not Sanger sequencing, which is what we've been using. So I'll leave it to perhaps yourself, Tom, to initiate that. That would be really good.
Excellent. Thanks, Tom. Okay, another question from Vanessa Ryan. Do you suspect that the Ammoniters have the pendulose ring, are a different species to the one that sticks out?
Yes, I think they are. I would definitely think this is the case. There are at least 2 species in Western Australia and probably more. There may be more widespread. I don't know, but this project may be able to indicate that. So we haven't sort of put all the data together yet. And when we do, we might be able to see whether we've got pan-Australian species or whether we have some which are restricted to the southwest of Western Australia. But that will happen in time. But it's really useful if there's a field character like the differences in the ring that you can use, rather than saying, well, you've got to look at the spores and see how big they are, or what shape they are before you can start to put a name to the species.
Yeah, carrying iodine and a microscope in the field is not always possible. So excellent. If there's any other questions, now's your chance. Yeah, that was really fantastic. Thank you so much, Elaine. I think we will go on now to hear from Rebecca. So PhD Rebecca Webb is from James Cook University. Beck is a research assistant in the 3rd year of her PhD. She has over a decade of experience in research on deadly amphibian chytrid pathogens, covering fungal culture, molecular biology, and frog exposure trials. Her project on glutathione biosynthesis in this fungus aims to understand how chytrid fungi manage to evade the diverse immune systems of a broad range of hosts. And this might hold the key to managing this devastating disease. As with our previous talk, you can use the Q&A function at the bottom of the screen to ask questions at any time. And if you've got another burning question for Elaine, you can ask that at the end as well. So Beck, whenever you're ready, go ahead and share your screen.
Is that working?
Yes,
Yeah,
all working.
cool. All right, awesome. Thank you guys for listening to my talk about glutathione biosynthesis in the amphibian chytrid fungus. Oh, well, why won't this work? Oh, here we go. So first of all, I want to start with a little game. So see if you can pick the odd one out. Is it 1, 2, 3, 4, 5, or 6? So they're all chytrids, which are an early divergent group of fungi characterized by their unwalled flagellated zoospores. And most chytrids are saprobes, but there are a few species that are parasitic that infect plants or algae. But there's only one genus that infects vertebrates, and that is Batrachochytridium. So Batrachochytridium infects amphibians and it causes the disease Ketomycosis. So if you haven't heard of Ketomycosis, the story starts right here in North Queensland, when in the early 90s, wildlife managers became concerned about the alarming disappearance of several different frog species from several different rainforests. And this graph here just shows the very dramatic decline of 4 completely different frog species from the same creek over the space of just 1 year. And at first they thought the cause might have been a virus, but a PhD student here at JCU figured out that the frogs were actually dying from a fungal disease, which she termed Ketomycosis. And this disease is characterized by skin shedding and redness, lethargy, and eventual death of the infected animal. The following year, the fungus was described. It was Batrachochytridium dendrobatidis, or just BD or chytrid for short. We now know that there are actually 2 species in this genus. There's BD, which infects mostly frogs, and recently discovered B. sal, which so far only infects a few salamander species. Today, I'm just going to be talking about BD. So the life cycle is similar to that of other chytrid species. It starts off with a motile zoospore, which develops into a zoosporangium, which then asexually produces more zoospores, which are released via a discharge tube. When it infects the host, it actually only occurs in the very outer keratinized layer of the frog's skin. So just this thin layer here, or in the keratinized mouthparts of tadpoles. In the 30 years since its discovery, we now know that this disease has spread all over the world and it has a huge host range. So it's caused severe declines in over 500 different amphibian species, and this includes the extinction of at least 90 species. So it is the worst wildlife disease ever. And Australia has been hit particularly hard. We have 6 species already extinct because of this disease, and several more at risk if we don't intervene. And this disease is not a thing of the past. There are ongoing outbreaks. So in this year, we saw particularly bad outbreaks all on the East Coast. So you might be wondering how this disease actually kills frogs, especially since it only seems to be a very superficial infection occurring in those very outer layer of the frog's skin. Well, the fungus builds up to the point where it damages the skin so that it can no longer transport electrolytes, and this results in electrolyte depletion, especially in regard to potassium and sodium. This electrolyte depletion then results in loss of homeostasis, and the frogs eventually die of cardiac arrest. And this mechanism might explain why so many different types of frogs all suffer the same fate, because the skin is such an important organ for frogs. But how does it get to this point? How does the fungi build up to such levels that the skin is no longer able to function? And how are they evading the host's immune system? Well, one way that other pathogens cope with the host immune system is via glutathione. So glutathione is a thiol tripeptide antioxidant, and it can exist in both a reduced or oxidized state. And it's pretty much universally important for normal cellular function because it maintains redox balance. But it's also really important for pathogens because it protects them from the oxidative stress generated by host immune cells. So phagocytic immune cells can use hydrogen peroxide to kill pathogens, but glutathione can neutralize the hydrogen peroxide and therefore protect the pathogen from the host. So the synthesis of glutathione starts off with this first enzyme, GCL, here in green, which joins the first two peptides, and then a second enzyme adds a third peptide to make the tripeptide glutathione. This enzyme here, GPX, is responsible for neutralizing the hydrogen peroxide, and in the process, glutathione is oxidized, and then this enzyme here, GR in yellow, can recycle the glutathione back into the reduced form so it can be used again. This diagram just illustrates how important the glutathione system is for fungal pathogens. Because if you knock out any one of these enzymes, you will create mutants that either have reduced virulence or reduced tolerance of hydrogen peroxide in a whole range of different fungi species. So my question is glutathione important for the amphibian chytrid fungus? And I'm going to be looking at two aspects. I'm looking at the expression of glutathione genes during cell development, and then also the consequences of depleting glutathione and then exposing the cells to stress. So starting off with glutathione expression, I'm looking at this first enzyme here, GCL, and I want to quantify GCL expression during early zoosporangia development. So that's when the fungus has just switched to the parasitic life stage. To do this, it's important to start with zoospores that are all the same age, and then use them to grow a lab culture or to infect a host. And then harvest messenger RNA at these various time points, measure it using PCR to see if this gene is differentially expressed over these different developmental stages. So the cutoff for differential expression is supposedly fold change of 1.5, and my results were much less than this, which indicates that GCL expression is fairly stable during initial sporangia development for cultures grown in the lab. But what about the fungus inside the host? Well, I did have some difficulties extracting pathogen messenger RNA from host tissues, which does make it a little bit difficult to make comparisons, but this also appears fairly stable. And at the very least, I can say that expression of this gene is not drastically upregulated when it occurs in the host versus when it is grown in culture. So overall, expression of the glutathione synthesis gene does not appear to change as the sporangia develops. And I was surprised by this result because I assumed that the expression would be upregulated in the parasitic life stage or when the fungus is occurring in the host. So next up, I wanted to see how important glutathione is during stress. And there is actually a chemical called BSO, which can inhibit this enzyme and therefore prevent the production of BSO. And we can use BSO to deduce the importance of glutathione. But I did not know whether BSO actually works on chytrid cells. So according to the literature, BSO should inhibit the glutathione synthesis in other fungi species at concentrations between 1 and 5 millimolar. So I tested it at a range of concentrations up to 30 millimolar, and then after 20 hours, I measured total glutathione levels using a commercial luminescence kit from Promega. And I found that BSO definitely did work. It depleted glutathione by up to 70%, but I needed much higher concentrations than that suggested by the literature. So now I know that BSO can inhibit GCL and prevent the synthesis of glutathione. That theoretically should mean the cells are less tolerant of hydrogen peroxide, including hydrogen peroxide generated by host immune cells. And this should mean that they are less virulent. So to test this, I tried to find out whether BSO treatment makes chytrid cells more sensitive to oxidative stress. To do this, I started with my chytrid cells, added BSO, waited 6 hours, and then added the hydrogen peroxide, and then measured growth a few days later. And to my surprise, I found that BSO did not increase sensitivity to hydrogen peroxide. So if we look at this blue line here, apart from a little bit of variation at 5 millimolar, adding BSO didn't decrease growth in cells that were exposed to oxidative stress. But glutathione does have other roles apart from neutralizing oxidative stress, which is related to avoiding the host immune system. It is also important for other things that might not be directly related to pathogen virulence. For example, detoxification of toxic endogenous metabolites such as formaldehyde and the sequestration of heavy metals. So I repeated these experiments and instead exposed the cells to formaldehyde and the heavy metal cadmium. Once again, I found that BSO did not increase sensitivity to formaldehyde, but I did find that BSO did increase sensitivity to cadmium. So you can see, once again, looking at the blue line, before any BSO is added, cadmium basically doesn't have any effect. But as soon as we add BSO, growth drastically decreases. So the next question is why? And the answer is, I don't know, but I suspect that when hydrogen peroxide is neutralized, it involves this oxidation of glutathione, but the oxidized glutathione can be reduced back and recycled and used again. So a fresh source of glutathione might not necessarily be required. But when it comes to cadmium, the glutathione is bound up and that cadmium is lost. So it does actually require fresh glutathione to replace it. So in summary, the glutathione enzyme can be inhibited by BSO to prevent glutathione synthesis. And this is the first time this is demonstrated for BD or for any chytrid species as far as I'm aware. But BSO doesn't increase sensitivity to oxidative stress. This might mean that the GCL enzyme is not that important for pathogen virulence in this species. So I'm not saying that glutathione isn't important full stop, but just in the short term, these cells, when they're under oxidative stress, seem to be able to manage without the production of fresh glutathione. But BSO does increase sensitivity to the heavy metal cadmium. And this might be an interesting topic for future work. So what's next for me? Well, the fact that the GCL expression is fairly stable over the life cycle and that we have a clear phenotypic effect in relation to cadmium tolerance might mean that glutathione is a good target for gene knockdown studies, which is the other part of my PhD, but I haven't had time to talk about today. So circling back to our original question, the Bacteroideum genus is definitely the odd one out. So out of all the chytrid species, they're the only ones that have switched to parasitizing vertebrates. But unlike other fungal pathogens, they aren't reliant on glutathione synthesis to tolerate oxidative stress. And this just highlights how flexible these funny little fungi are and might shed some light on why they are such successful pathogens. Thank you.
Thank you so much, Beck. Just picture a virtual round of applause. You can put your video on if you want to.
Sure.
Yeah, no, that was fascinating. Thank you so much. I have two questions I'm going to kick off with. First of all, you mentioned that there is another chytrid fungus and that infects salamanders. We don't have native salamanders in Australia, as far as I know.
No.
Is that chytrid fungus present here that you know of?
No, it's not. It's only present in Europe so far. So they are really worried about it getting into the States because they have a lot of salamander diversity. But the B cell can actually in the lab infect frogs. So we do need to be careful that we don't introduce it into Australia as well.
Yeah, okay. And now that I mean, this may be part of the next stage of your PhD, but now that you know that BSO affects, has a correlation with cadmium, is there a way to apply that in the field? Is BSO something that has an application for prevention of infection?
It definitely highlights a weakness in the fungi for sure. So if there were, say, antifungal drugs that could target glutathione synthesis, definitely, yeah. But that's a little bit beyond the scope for me, but it's a start.
Yeah, okay, excellent. So Joanna Wong has a question. Firstly, poor frogs, she says.
Yeah.
Do you know of any other frog species that are actually resistant to BD infection?
Yeah, and that is, so there's one called Crunia signifera, which seems to be, I think it's actually called tolerant, so it can get infected, but doesn't seem to build up to any levels to kill the frog. And they have no idea why. That's a really interesting part of, research that people are trying to figure out.
Yeah, I imagine if there's some avirulence resistance gene work done on that, it'll be very interesting to.
They are, yeah, they're looking at resistance genes, yeah.
Yeah, okay. And there's also a new effort now to see if you can use RNAi in these kinds of pathogen controls. So
yeah, that's what I'm trying to do.
Oh, yep. Cool. That sounds really fascinating. Okay, yeah, Joanna says thanks. That's really interesting. And another comment from Vanessa for Elaine. Thank you. Having an obvious feature for identifying Ammoniter would be very useful. D. Carter has a question for Beck. Aren't cane toads also resistant?
Yeah, small cane toads can definitely become infected and die. So it seems to be dependent on how old they are. So they're not necessarily, I don't know what it is that's changing when the cane toad reaches a certain age. It does somehow seem to switch to being more resistant to the fungus.
Oh, interesting. Okay. Good. All right. Are there any other questions from anybody? No one's got their hands up, but yeah, thank you so much. Really great to see the progress and looking forward to see what comes out the next stage of your research for both of you. As I said, we're hoping to have our conference in New Zealand next year, and it'd be great to have you there. Teach us more about what you learnt with the support of the grant. So thank you to both of you for joining us today. And also thanks to everyone else for being here to listen to their fascinating research. Please take a moment to answer the quick survey. And don't forget, if you have research to share, get in touch with me and we can sign you up to present your work during our 2022 seminar series. But for now, it's December already, so happy holidays, and hopefully we'll see you next year. Thanks very much, everyone. Bye.